Hello, my dear students. Welcome to my today's online class on continuation of the understanding of the phenomenon of fertilization. In my previous class, I had taught you the definition, the types of fertilization, and part of mechanism. We had understood in our previous interaction the first step pertaining to the mechanism of fertilization. Today we shall continue with the second step and third step, understand it, and then we shall understand the significance of fertilization. All right. So before getting into the second step in understanding the mechanism of fertilization, I just want to briefly pass through the PPTs what we have learnt yesterday. So this is the introduction PPT, right? I'll just roughly rush through this introduction part, definition, types, internal fertilization, external fertilization, right? These are all the things what we discussed yesterday. Prerequisite of fertilization, then state of ovum and sperm before fertilization, right? Then, so steps of in fertilization, penetration, what we learned yesterday, activation today we are going to discuss, and fusion of gametes also we are going to discuss. So physical process of penetration, all this fertilizing, anti-fertilizing reaction in sea urchin and later found in other organisms also. Then different views, and then what exactly happens during penetration. These are all some of the things what we discussed in detail yesterday. So then the chemical processes involved, and then precursor of active principles, concept of androgametes and gynogametes, two types of gynogametes and three types of androgametes. Then, so in other than these gametes, the other chemicals what are needed for penetration to take place. Then, so how exactly the equilibrium of androgamete and gynogamete bring about successful penetration and positive hemotaxis, right? So I have given a detailed account in my previous lecture of these PPTs. Now let us study the next step in fertilization, that is activation of fertilization. So you can see the diagram here. You find the endoplasm, ectoplasm, and that is surrounded by the plasma membrane. Now it is called oolemma. Oolemma is surrounded by another egg membrane called vitelline membrane, and you will see the cortical granules, right, in the cortical region, black colored ones. One half of it is actually the status of the egg, plasma membrane, vitelline membrane and cortical granules prior to penetration, that is in an unpenetrated egg. And the other half will show you what exactly happens once the sperm penetrates the egg and how exactly fertilization is activated. You can see that the vitelline membrane lifts itself apart. So the cortical granules move, okay, and happen to lie below the vitelline membrane. Lot of perivitelline space is created. So now there is lot of space between oolemma and vitelline membrane, and cortical granules will shift right? And they swell, and now the vitelline membrane becomes very thick, and now it is called fertilization membrane. What are the events that happens systematically? I'll let you know little later. Right now, but keep this diagram in your mind. Now we shall understand the activation process of fertilization. Okay. Now, what exactly you mean by activation is, it is something like awakening of the ovum or egg, okay, from its deep sleep into the state of activity, right? So let us see what happens now. So now, though many changes in the penetrated egg are known, people have studied, but still a clear understanding of exactly what happens, what biochemical events take place, which bring about necessary stimuli to activate the egg, is not clearly understood even today. Even though there is lot of documentation. All right. Then let us move to the next PPT. Same diagram I have posted. Now, so the first event of activation involves cortical reaction, what I was telling, and I'll talk about this cortical reaction little later when I talk about polyspermy and prevention of polyspermy. All right. Now, just you understand that the first event of activation is the cortical reaction, that is formation of fertilization membrane, what I have explained in brief. After telling you few more points about activation of ovum, I'll come back to this concept again. So the vitelline membrane lifts itself from the cortical surface. You can see in the right half of the diagram to form a large space called perivitelline space between ulama and vitelline membrane. Then subsequently, dense lamellar materials, which are the derivatives of cortical granules, which are black colored one because of black pigments, are accumulated towards the inner side of the vitelline membrane, and now with the thickening, now it is no more called vitelline membrane. Now we call this as fertilization membrane. Okay. So first one is lifting up of vitelline membrane. Okay. And formation of a large perivitelline space. Then accumulation of these lamellar materials, which are derivatives of cortical granules, towards the inner side of the vitelline membrane, now called fertilization membrane. Okay. Now, I'll come to this concept a little later. Now we shall understand structural changes. What you see in case of penetrated egg. Penetrated egg, I mean when the sperm has penetrated into the egg. First change. Cytoplasm moves in streamlined fashion. Cytoplasm moves in streamlined fashion towards the point of sperm attachment. At different components of cytoplasm are reshuffled and reorganized. And some irregular amoeboid movements also result into the final return to the spherical shape. The refractive index, that is mu, what you have studied in your PU classes, a physics concept, alters. The refractive index mu of the cortex alters. Then thickness of the membrane increases. The cytoplasmic part or the vitellus reduces in volume. And now I should tell you, this vitellus is nothing but the reserve food material or the yolk, and there is considerable decrease in the volume of yolk. Alright. Then there is redistribution of calcium in cytoplasm, resulting in the change of viscosity of the cytoplasmic part of the cortex. Okay, so redistribution of calcium in the cytoplasm resulting in the change of viscosity in the cytoplasmic part of the cortex. Very important change to be noted. Then near the region of sperm attachment, a small pseudopodium or cone-like structure is formed which engulfs the sperm head, and this is called fertilization core. Okay, and earlier people thought that the formation of fertilization cone is seen only in echinoderm eggs, but with understanding with recent electronic microscopic analysis, we have understood that it happens in most of the eggs, including mammalian eggs also. So these are the structural changes to list in a penetrated egg. Okay. After this, you have to learn one more concept. What are metabolic changes that you see in a penetrated egg? Now, first one. See, in an unpenetrated egg, means when the egg is still in its sleeping state, there is an enzyme, there is a cytochrome oxidase inhibitor. Okay. Now, which is inactivated after fertilization. Once the sperm enters, this particular chemical inhibitor is inactivated. Therefore, now after penetration, the egg takes lots of quantities of oxygen. This results in increased uptake of oxygen, okay, probably needed for various metabolic activities. First change. Then secondly, a mature unpenetrated egg, has little ability to incorporate amino acids to protein. It has been found that in spite of this disability, the enzymes isolated from such an egg do synthesize protein in vitro. That is, in vitro means laboratory conditions. Okay. In vivo incapacitation of protein synthesis is believed to be due to suppression of a functional state of mRNA or ribosomes. The moment the sperm penetrates, this inhibition is removed, and as a result, the ovum starts synthesizing proteins. So there is synthesis of proteins. Okay, there is uptake of more oxygen. There is synthesis of DNA. Okay, so DNA synthesis resumes after penetration. These are the three metabolic changes that take place in a penetrated egg. So after understanding the structural changes, then metabolic changes, now we should understand one more concept, resumption or beginning of meiosis, that is gametic meiosis. Okay. See, when oogenesis is taking place, that is formation of ova in ovary, for necessary organization of the ovum, meiosis remains suspended at various stages in different groups of animals. So meiosis remains suspended, okay, excepting sea urchins, sea lantrates, where meiosis is independent of fertilization. But in all other animals, meiosis remains suspended, and now there is resumption of meiosis. Okay. Now, what happens in which group of animals we will see? No meiosis occurs before fertilization in case of ascaris, that is Janthula, mollusks and crustaceans. One category. Second category, the meiosis is arrested at the metaphase of first meiotic division in case of insects and nematines. Then, third category, arrested at the end of meiotic division For example, amphibians. Okay. So like this, in different organisms, the phase of arrest differs. But whatever may be the cause of arrest, now it continues and meiosis ends during the period of activation. Very important concept to note down. Okay. Now, one more thing simultaneously happens. Block to polyspermy. This physiological polyspermy, pathological polyspermy, and different mechanisms to bring about the prevention of polyspermy in monospermic animals I will discuss in my next video. But right now, in brief, I will tell you, because without this we cannot proceed further. So in most of the eggs, considerable changes take place during activation, by which entry of more than one sperm is prevented. Okay. In the eggs of annelid, sea urchin, starfish, amphioxus and frog, it is done by the formation of a fertilization membrane, what we call cortical reaction. Okay. Cortical reaction or slow block mechanism. I'll come to this little later in my next video. So, one more, efforts are being made to block polyspermy and to see that only one sperm fertilizes the ovum. Okay. Then, yes, I was telling you, I am going to present few more points regarding formation of fertilization membrane. The diagrams what you have seen in slide number 23 and 24. So in an unpenetrated sea urchin eggs, the vitelline membrane is thin and it is closely set with plasma membrane, right? We have seen that in one half of the egg, which exhibits the state of unpenetrated egg. Within the cortex, the cortical granules remain enclosed in vesicles. Okay. But immediately after penetration, the cortical granules are released from vesicles and these contents adhere to the inner surface of the vitelline membrane. That's what I was telling you. Then the vitelline membrane lifts up from the plasma membrane, and this lifting is accompanied by accumulation of fluid. Okay, accumulation of fluid and a hyaline layer between vitelline membrane and plasma membrane. Now the vitelline membrane becomes thicker and it is called fertilization membrane. Okay, now let us see now. Runstone has shown that this fertilization membrane is formed by separation of cortical and pigment granules. Cortical and pigment granules. Okay. Then, the eggs of teleostean fishes. Here, a little deviation is seen. Very interesting, you should know. The eggs of teleostean fishes have a hard covering called chorion. Okay. And there is a small opening called micropyle, which serves as passage through which only one sperm can enter at a time. Okay. Rest of the surface is covered by the calcareous chorion. Chorion, not to be confused with amnion allantois chorion. That chorion is different. This chorion is different. And immediately after the entry of one sperm through the micropyle, the cytoplasm of egg invades to close the micropyle. This is a special adaptation seen in teleostean eggs, okay, to prevent polyspermy. Okay, then in mammalian eggs, what happens? Let us see. The prevention of entry of more than one sperm is done by progressive changes in the zona pellucida, which is known as zona reaction. In other animals, you call it, in amphibians, say for example, you call it cortical reaction. And in mammals, surrounding the ulemma or the plasma membrane, you find one more layer called zona pellucida. Okay, And there are changes, progressive changes in this layer of zona pellucida once the sperm makes its entry, okay? And this is known as zona reaction, and this zona reaction takes care of prevention of the entry of more than one sperm. So different groups of animals have evolved their own ways and means to prevent polyspermy, where monospermy is a rule, okay, to avoid in a way pathological polyspermy, okay. Now in the eggs of many other organisms like insects, elasmobranchs, urodils, reptiles, birds, etc., one more deviation you see, more than one sperm may enter within the egg. More than one sperm may enter within the egg, but please note, only one male pronucleus unites with the female pronucleus. The other sperm pronuclei help in the breakdown of yolk. This is somewhat beautiful to understand, right? So, this is how the different groups of animals have found out their own ways and means to prevent polyspermy and to see to it that single male pronucleus fertilizes the female pronucleus. So this is the second step, that is activation of fertilization in understanding the mechanism of fertilization. Now with this background, we shall proceed to the next stage, that is fusion of pronuclei. Okay. Now the nucleus of ovum or the egg is called female pronuclei, which is bigger in size, and the nucleus of sperm is called male pronucleus. Okay. Now these two pronuclei should come together, and this process of male and female pronuclei fusing is called amphimixis, a very important concept to understand in the process of fertilization. Okay. Now fusion of pronuclei. Okay. Fusion of pronuclei. Now the process of fusion varies in different organisms. In some there is complete fusion. In others, the chromosomal elements arrange themselves along the spindle fibers. Okay. So one concept, there is complete fusion. In still others, the chromosomal elements simply arrange themselves along the spindle fibers. Formation of spindle fiber is a very important concept during cell division, right? So the process, what happens in case of amphibians, as exemplified by frog, is described here. Okay, we shall see that the sperm head and the middle piece enter within the egg cytoplasm. Immediately after the entry, the egg completes its meiosis and loses its centriole. Okay, this is the first step. Sperm and the middle piece enters. All right. This I am talking only about amphibians. In different groups, different parts. I mean, the sperm enters into the egg to different extents. Okay. Now, the second step. After the entrance, the sperm rotates through 180 degrees. Okay. Straight angle. After the entrance, the sperm rotates through 180 degrees and thus the middle part becomes the front end. And at this stage, the sperm moves within the egg at random. And this path is called penetration path. The diagram I will show you in my next PPT. Then, the egg pronucleus moves towards the periphery and the direction of the sperm becomes oriented to it. And the second path is called copulation path. Both the paths are trailed by marks of pigments. So you should remember about penetration path and copulation path. Later, when they fuse, they move further and that is called cleavage path. I'll show you the diagram. You will understand it better. Yeah, you can see here the polar bodies. You can see the penetration path, the sperm getting into and taking 180 degrees and moving towards the female pronucleus. Okay, that is the path of sperm pronucleus, male pronucleus. Then it moves towards the female pronucleus, that is copulation path. And then later, after fusion, it moves downwards and that is called cleavage path. Okay, now, so considerable changes occur within the sperm pronucleus. Now it swells up and the chromosomes become reticular in appearance. Reticular in the sense network-like in appearance. Then, sorry for the typographic error. Not in the end piece, in the mid piece. Actually, the centrioles or the centrosomes are present in the neck region only. Okay, not even in mid piece. This is not end piece. It should have been mid piece and above the mid piece, right? Okay, especially in the neck region of the sperm. In the neck region, we'll put it this way. In the neck region, the centrosome and the centriole form the asters around them. All right. Then, the centriole of the sperm divides and moves to the poles, forming the fibers in between them, where the chromosomes of male and female pronuclei arrange themselves in the equator. After the disappearance of nuclear membrane, by now the nuclear membranes in both would have got disappeared. All right. Then, so it resembles metaphase plates of mitosis and soon results into cleavage. Okay. So this is what exactly happens during amphimixis or the fusion of male and female pronuclei. With this, dear students, we have understood all the three steps in the mechanism of fertilization. Once we are done with this, it is the time to understand the significance of fertilization. There are three significances which you need to learn. Okay. Now, first significance, it ensures propagation. Okay. It ensures propagation. So, the union of haploid chromosomes and the attainment of diploid state help the organism to propagate its own species. Right? That is restoration of diploidy, what we say. Then in lower organisms, the entire individual is involved, but in higher forms, this is achieved through sexual reproduction and by the fusion of two specialized cells. Then yet in another few other organisms like earthworms, planarians, etc., restoration of normal chromosome number occurs in a variety of ways. See, what happens here is the chromosome number would have got doubled just prior to meiosis, and the meiotic division causes the adjustment of normal chromosome number. Okay. So, this is the significance of fertilization. Then second one is rejuvenation. The union of two cells provides some stimuli which are necessary for the vitality of the individual. So, rejuvenation is another significance of fertilization. More than anything else, it produces variations. Alright, fertilization brings about variations, we'll see. The process of gametogenesis, results in the production of gametes which are dissimilar among themselves and to their parents. Okay, because exchange of genetic material takes place during the Packetin stage between non-sister chromatids of homologous chromosomes. If you have studied meiosis in detail, especially the first meiosis, you will understand this concept better. So, as a result, the gametes are dissimilar among themselves. Gamete A is not same as gamete B, though they are the products of same gametogenesis, and they do differ from their parents. Then, so The recombination of these gametes therefore produces large number of variations which face the ordeal of natural selection. Okay. Fertilization thus ensures the production of variations. So these are the three significances which you need to understand. Okay. Significance of fertilization. Okay. Now, yes, dear students, I have given two YouTube links here. Okay. It is better you watch these, one for 5 minutes and one for 3 minutes. So these videos will tell you how exactly fertilization takes place in humans, and one of the videos how exactly fertilization takes place in animals. Better you watch these two videos along with whatever I have taught you. Okay, that gives you a better understanding of the mechanism of fertilization. So with this, I complete the second video lecture on fertilization. Second video lecture on fertilization. So, see you in my next class to talk about the polyspermy. Alright? Till then, stay blessed, study well,